Welcome and thank you for joining us today for this introduction to microRNA. I will be covering a broad range of topics with the aim of conveying the essentials of how microRNA was discovered, its biogenesis, its normal role in cells, and evidence that misregulation is involved in disease. I'll then switch gears and tell you how to approach the analysis of microRNA, including methodologies for isolation and quantification, and tools for profiling changes in microRNA expression. We'll end with a brief description of some of the tools available for manipulating the levels of functional microRNA in cells in an attempt to establish a relationship between microRNA, target, and function. This webinar is the first of a series of 3 microRNA-related webinars. The other 2 webinars will also be given by colleagues of mine at QIAGEN. The second webinar will focus on experimental design and data analysis of microRNA-related studies. And the third webinar will focus on the usefulness of microRNA expression profiling in cells, FFPE samples, and serum to aid in biomarker development. At the end of today's webinar, please feel free to ask any questions. Most of you probably know the early stories of how RNAi was discovered. In a wide range of organisms, peculiar phenotypes of gene suppression were observed that had no ready explanation. The breakthrough in understanding came from the work of Andy Fire and Craig Mello, who were working with antisense RNA in C. elegans in an attempt to knock out the function of genes. They discovered that a minor contaminant in their antisense RNA prep was in fact a very potent silencing molecule. This contaminant was double-stranded RNA, and led to their discovery that double-stranded RNA could direct the silencing of a gene. The mechanism for this gene-specific silencing, as it turns out, is conserved from plants to humans. The use of RNAi to the organism in question varies from defense against viruses to the fine control of gene expression, which we will discuss later in this talk. There was an intense amount of interest in the science of RNAi, but also on the economic potential as a boon for the pharmaceutical industry. It's now 8 years later, and while there are clinical trials underway, it has proven very difficult to translate the promise of RNAi into blockbuster drugs. So let's begin with the canonical pathway of microRNA biogenesis. Mature microRNAs are first transcribed by RNA polymerase II as a long primary transcript, or primary microRNAs, which may contain more than one microRNA. In the nucleus, primary microRNAs are then processed to hairpin-like pre-miRNAs by RNase III-like enzyme DROSHA. Pre-miRNAs are then exported to the cytosol by exportin 5, where they are further processed by the RNase III-like dicer from precursors into mature microRNAs. These microRNAs are then incorporated into RISC, and microRNAs with high homology to the target mRNA lead to mRNA cleavage, and microRNAs with imperfect base pairing to the target mRNA lead to translational repression and/or mRNA degradation. A highly conserved microRNA family has about 300 conserved messenger RNA targets in mammalian systems. Most mammalian protein coding genes are under selective pressure to maintain 3' UTR pairing to the seed of one or more microRNAs. The 5' region of the microRNA is the most conserved portion. The 5' region, particularly from nucleotides 2 through 7, base pair with the target sites in the 3' UTR of the mRNA by Watson-Crick pairing, and this region of the microRNA is called the seed sequence. The first nucleotide is not required to base pair, and there is a preference for A on the target at this position. And a non-U is preferred at the nucleotide position 1 on the microRNA. Shown are the canonical base pairing models for the seed regions of a microRNA to an mRNA target, with a seed match 6-mer being the weakest when compared to an 8-mer match. A number of different kinds of studies have taught us about how a microRNA guides risk to recognize an mRNA target, including whole transcriptome experiments, reporter gene constructs containing binding sites, enrichment of bound targets by pull-down assays, and sequencing and alignment of putative binding sites from different species. Clearly, the largest predictor of an interaction and the one with the best experimental evidence is the match of the microRNA seed region with the target in the 3' UTR of an mRNA. All this data together is used to develop target prediction algorithms, which have varying degrees of overlap with each other. I would now like to turn to describing how the loci which encode microRNA are organized and controlled, and how microRNA diversity is generated by transcription and post-transcriptional levels. Genomic organization of microRNA encoding genes is quite varied. Intergenic microRNA genes are found in genomic regions distinct from known transcription units. These microRNAs can be monocistronic with their own promoters, or polycistronic, where several microRNAs are transcribed as cluster of primary transcripts with a shared promoter. Intronic microRNA genes are found in the introns of annotated genes, both protein coding and non-coding. These microRNAs can be present as a single microRNA or as a cluster of several microRNAs. Intronic microRNAs are thought to be transcribed from the same promoter as the host gene and processed from the introns of host gene transcripts. Exonic microRNAs are quite rare compared to either of the types above and are often overlap an exon and an intron of a non-coding gene. These microRNAs are also thought to be transcribed by their host gene promoter, and their maturation often excludes host gene function. There are many examples where a particular mature microRNA is generated from multiple genomic loci. As can be seen for miR1302, there are 11 genomic loci present on different chromosomes, and some are intergenic, some are intronic non-protein coding, some are intronic protein, and some are intronic protein coding. One can really appreciate the diversity and complex regulation of expression of this mature microRNA, considering that each locus will be under different promoter control. In addition, there are other ways to generate diversity in the micronome. Deep sequencing reveals polymorphisms generated by both a bit of wobble in the processing and by non-templated addition of bases to the 3' end. Interestingly, these modifications aren't always random, but are situation dependent. They also play havoc with RT systems that depend on a perfect match at the 3' end for priming. Using miR21 as an example, note the 3' polymorphisms. In fact, the minor component marked by an arrow is actually the sequence listed for miR21 in miRBase. There are also reports of adenosine deaminase RNA-specific, or ADAR editing, on microRNAs, which further increases diversity. ADAR destabilizes double-stranded RNA through conversion of adenosine to inosine and generates diversity by blocking Drosha processing, blocking dicer processing, and also changing the recognition of the microRNA for their targets. Now I'd like to turn our discussion to what happens when things go wrong. As exciting as it is to discover the role of microRNAs in normal human cells, the emerging understanding of the role of microRNA in disease is truly remarkable. But you could already imagine if something that is essential for normal cellular processes is disrupted, cells will behave badly. So how would a microRNA contribute to disease? Well, some of these were discussed already, such as the potential effect of a SNP or variability in processing. Of course, changes in the regulation of a microRNA, either through methylation of the promoter or changes in the expression of a transcription factor that regulates the gene, can have an effect on microRNA levels. Also, genomic instability, either through amplification or deletion, translocation, or insertional mutagenesis, would drastically change the expression levels of a microRNA. Further, changes to Drosha and dicer processing can disrupt normal microRNA function. Looking back a few years to the connection of microRNAs to disease, in 2005, results of array analysis of microRNA in cancer suggested that there were specific microRNA signatures associated with human diseases. Specifically, microRNAs located in genomic regions amplified in cancers can function as oncogenes, whereas microRNAs located in portions of chromosomes deleted in cancer can function as tumor suppressor. Furthermore, abnormal expression of microRNAs had been found in both solid and hematopoietic tumors, and microRNA expression fingerprints were determined to correlate with clinical and biological characteristics of tumors. And truly, the race was on to develop microRNA biomarker therapeutics. Flashing forward to present day, many studies point to misregulation or misexpression of microRNAs in many cellular processes that are fundamental to oncogenesis in a variety of tumors. One can imagine that as time goes on, the role of microRNA in diseases will continue to be elucidated, necessitating products that enable high-quality microRNA analysis. Now I'd like to turn to speaking about methodologies for microRNA analysis. As a first step in the analysis of microRNA, we need to isolate microRNA, and QIAGEN is naturally a place to turn for this technology. QIAGEN offers the microRNAs preparation kit that permits effective purification of total RNA, including microRNA, from a broad range of cells and tissues. And the workflow is shown here on the left, where you first lyse and homogenize your cells or tissues. You add chloroform and separate the phases. And you can purify microRNA and total RNA together. Also, too, the kit allows for efficient enrichment of microRNAs, where you can also purify the microRNA fraction separately from the total RNA. And also an important point to note is that this spin column format has been automated on the QIAcube for walk-away microRNA purification. And there is a 96-well format that can be automated on larger QIAGEN sample handling robots. And regardless of whether you choose to do a total RNA enrichment or separate fractions of microRNA from the larger RNA, you have high purity RNA that is suitable for sensitive downstream applications. In addition, QIAGEN also offers the microRNAs FFPE kit, which is ideal for purification of microRNA from archival samples. And this is very important, as there are over 500 million FFPE tissues that are archived, and the discovery of importance of microRNAs is very well spread. And traditionally, there are problems with isolating RNA from FFPE tissues, that the RNA quality is typically heavily fragmented, and cross-linking by formaldehyde can interfere with the RT. In addition, with traditional methods, the RNA yield is normally low, and the procedure may actually not purify all usable RNA. The procedures may also increase fragmentation, and they're often ineffective in breaking up cross-linking. And this is where the microRNAs FFPE kit stands head and shoulders above. This is a novel method that prevents cross-linked RNA from blocking downstream application. There's an optimized lysis buffer with proteinase K, and also there is a procedure for, it is a RNAs mini-lute column for small elution volumes. In addition, we also offer a solution for purifying total RNA, including small RNA, from serum or plasma samples. And this is a supplementary protocol associated with the microRNAs mini kit. And this kit is also very useful for preparing total RNA for use with the miScript microRNA PCR array, which I'll discuss later in my talk. Now I'd like to shift gears and talk about microRNA quantification technologies. Here I'd like to discuss the miScript PCR system. This is a brand new system recently released by us at QIAGEN, and the miScript 2 RT kit has a dual buffer technology that offers both exquisite specificity and flexibility. The first buffer I'd like to talk about is the HiFlex Buffer in the microScript 2RT kit. What this buffer allows is specific detection of mature microRNAs. And you want to use this when only mature microRNA detection is desired in your experimental studies. And this includes when you want to do single microScript primer assays for mature microRNAs, or when you want to do either pathway-focused profiling using microScript microRNA PCR arrays, or whole micronome profiling using micronome microScript microRNA PCR arrays. The second buffer found in the microScript 2RT kit is the microScript HiFlex Buffer. And what this buffer allows is unparalleled flexibility to detect all RNA molecules from a single cDNA preparation. And this includes mature microRNAs, precursor microRNAs, mRNAs, and other non-coding RNAs. And this buffer is ideally used when mRNAs, precursor, or other long non-coding RNAs need to be quantified in parallel with mature microRNAs. To give you a little bit of background of how the reverse transcription reaction works, that in the HiFlex buffer formulation, microRNAs are first polyadenylated and reverse transcribed in a single tube reaction. And microRNAs are first polyadenylated because microRNAs naturally do not contain a poly-A stretch in nature. Once the reverse cDNA is prepared, you then use a microScript forward primer assay that is specific for your mature microRNA of interest in conjunction with your microScript universal reverse primer to amplify your mature microRNA of interest. In HiFlex Buffer, in addition to having the same mechanism to amplify mature microRNA, random primers are also included that in conjunction with the oligo-DT primer, larger RNAs, such as mRNAs, are reverse transcribed, and then using a Quantitech-specific forward primer and Quantitech-specific reverse primer, your mRNA of interest is amplified in your qPCR reaction. And regardless of whether you choose the HiFlex or HiFlex buffer, you have the same streamlined workflow. You simply mix your RNA, your either HiFlex or HiFlex buffer, water, and a 10x nucleics mix, and your reverse transcriptase enzyme mix. You incubate your RT reaction at 37 degrees for 60 minutes, followed by heat inactivation at 95 degrees for 5 minutes, and then use this cDNA to set up real-time PCR reactions. And as an example of when to use the HiFlex buffer, and this is, again, where you want to detect microRNAs and mRNAs from the same cDNA sample. And HiFlex buffer should be used to prepare cDNA for the quantification of these RNA species using the appropriate primer assays. And shown here, from a single cDNA preparation, you can quantify both mRNAs and microRNAs from the same cDNA preparation. On the other hand, HiFlex buffer does not permit the reverse transcription of any longer RNAs, such as mRNAs, into cDNA. In the same experiment performed using HiFlex buffer, you can see that the mRNAs are not reverse transcribed, while the microRNAs are still reverse transcribed. And whichever format you need to achieve your experimental goals, the microScript 2RT kit offers robust performance. Shown here, either buffer enables exceptional linearity over 6 logs of input RNA. And on the x-axis, we show that a range of 10 picograms to more than 1 microgram of RNA in a reverse transcription reaction using either HiFlex or HiFlex buffer results in linear detection in qPCR. In addition, the microScript 2 PCR system offers exceptional sensitivity, and you can detect from only 10 copies to more than 1 million copies. And some background of this experiment was that a synthetic miR21 was reverse transcribed, and copy numbers from 10 to 1 million were used in a microScript PCR reaction. So now I'd like to turn to what you can do with the HiFlex Buffer. And as I mentioned previously, using the microScript 2 system with HiFlex, you can quantify mature microRNAs using microScript primer assays, small RNAs using custom assays, mRNAs using quantitative primer assays, and precursor microRNAs using microScript precursor assays. And just as a background of why you would want to quantify pre-miRNA stem loops, and you want to do this when you want to study transcriptional and or post-transcriptional regulation in microRNA biogenesis and its downstream implications. And also when you want to definitively identify which closely related genomic loci are expressed. And just to show how these assays work, that mature microRNA primer assays only detect mature microRNA, and these do not co-amplify precursor microRNA under extensive studies that we have done. And on the other hand, microScript precursor assays only amplify precursors. And the precursor assays are both a forward and specific pre-miRNA primer, and in extensive studies these have been shown to not co-amplify mature microRNAs. And just as a note, microScript precursor assays amplify all RNA species that contain the stem loop, including primary microRNAs and precursor microRNAs. And shown here is an example of the power of the HiFlex Buffer. It again allows parallel quantification of precursors and mature microRNAs from the same cDNA. And what we have here is the relative levels of various precursor and mature microRNAs in HeLa cells. And I want to direct your attention to a couple bars on the graph, including LET7A. The LET7A mature sequence is actually derived from 3 separate precursor microRNAs. And you can see that these precursor microRNAs contribute differentially to the final mature microRNA product. I'd now like to turn to microRNA profiling technologies. And I'd like to introduce the microScript microRNA PCR arrays that have just been recently launched by QIAGEN. And these arrays offer complete micronome genome or micronome coverage. And we currently have cataloged species for human, mouse, rat, and dog. And we have more than 1,000 assays for human, nearly 1,000 for mouse, and 650 for rat, which is more than 95% of the rat micronome. And we also have nearly the entire dog micronome assays for the entire dog micronome. And in addition, if you do not want to do whole micronome profiling, you can also do pathway-focused profiling. We have more than 10 arrays for human, mouse, and rat. And we have an MI Finder array, which consists of the 84 most widely published microRNAs. We have a variety of cancer-focused arrays, cell differentiation and development, immunopathology, inflammatory response, neurological development and disease, and also an array dedicated for serum and plasma experiments. And what I'd like to mention is that every assay found on either the micronome or the pathway-focused arrays is extensively bench validated. So what this means is that when you do an experiment, that you can have complete confidence that if your microRNA is expressed, our assay will detect it. And to show you a little bit about what this array looks like, each PCR array contains dried primers, and in the case of a 96-well array, you have 84 focused microRNAs, and in the final row, you have 12 control wells. The first 2 wells are for the C. elegans miR39 microScript primer assay, and this is used as a spike-in control in conjunction with a serum and plasma supplementary protocol. The next 6 wells contain the microscript controls for normalization, and the final 4 wells contain the microRTC and the PPC, which are used to assess reverse transcription efficiency and PCR efficiency, respectively. And a very nice feature about these arrays are that they are compatible with a wide variety of 96 and 384 well instruments, including instruments from Applied Biosystems, Eppendorf, Stratagene, Biorad, Roche, Takara, and QIAGEN. And in addition, that if you do not have the resources to do profiling on your own, we have a PCR array service core here at QIAGEN that can do everything from sample isolation to PCR array profiling to data analysis. And with the microScript microRNA PCR arrays, you have a very robust, straightforward protocol. You simply convert your microRNA to cDNA, combine your cDNA with Quantitech CyberGreen PCR master mix, and aliquot this across the microScript PCR array. You run this in your real-time PCR cycler, and you upload your raw CT values to our free data analysis tool that will automatically perform the delta-delta CT method of relative quantification and present your results in a variety of visual formats, such as scatter plots and volcano plots. And like the RT kit, the microScript PCR arrays offer robust and reproducible performance. On the left chart, you have a technical replicate 1 versus a technical replicate 2 of profiling of microRNA expression in HeLa S3 cells using an MI finder array. And the R-squared value is nearly 0.99 for a technical replicate. In the center graph, you have a biological replicate using HeLa S3 cell profiling versus a second biological replicate doing the same. And again, the R-squared is 0.99. And a very nice feature of these 2 studies was that these studies were performed by a first-time user of the system, and that really attests to how straightforward and easy the protocol is. In addition, on the right, we have that between 2 operators doing the identical experiment, the R-squared is nearly 1. And collectively, you can see that these arrays offer very robust and reproducible performance. As an application example of what you can do with these arrays, I'd like to show you an experiment we did where we did whole micronome profiling of HCT 116 colorectal cancer cells that were treated with or without the 5-AZA 2-deoxycytidine treatment, which irreversibly inhibits DNA methyltransferase-driven DNA methylation reactions by incorporating into DNA and covalently binding to the active site of the DNMT. A scatter plot of 2 to delta CT values demonstrates that significant differences exist in the mature microRNA expression levels of the 2 samples that were tested. Nearly 105 microRNAs were strongly upregulated in the treated cells, while only 30 were strongly downregulated in the treated cells. When a p-value of 0.05 is applied, the expression upregulation of 89 of the 104 microRNAs is significant, and the expression downregulation of 21 of the 30 microRNAs is significant. And we use the online microScript microRNA PCR data analysis tool to perform these calculations. And again, I'd just like to mention that this data analysis is free for you to use. I'd like to now finish with a discussion of microRNA functionalization technologies. And microRNA functionalization technologies involve manipulating microRNA function. And you can do this through either using mimics or inhibitors. And microScript microRNA mimics are synthetic double-stranded RNAs that mimic naturally occurring microRNAs after being transfected into cells. Once transfected into cells, synthetic mimics mock the endogenous activity of microRNAs, and what this does is that the mimic then binds to its mRNA target, and the mRNA is not translated, leading to a decrease in protein levels. In its counterpart, the miScript microRNA inhibitors are synthetic single-stranded chemically modified RNAs that specifically inhibit microRNA function after transfection into cells. And the function of the microRNA can be inhibited by the inhibitor binding, and this prevents processing, affects biogenesis, or can prevent translocation from the nucleus. And what this amounts to is that the microRNA cannot bind to its target mRNA, and what this leads to is that the mRNA is translated into protein. And the features of the miScript mimics are, again, that they are a double-stranded RNA oligonucleotide, and the functional sequence is the same as the naturally occurring mature microRNA. And transfection of microRNA, of the microRNA mimic, results in inhibition comparable to the endogenous microRNA, and the actions are stable. The actions of the mimic are stable in culture for up to 72 hours. And for this experiment, a miScript mimic for miR1 was transfected in the HeLa S3 cells, and at the indicated time points, cellular lysates were prepared, and the protein levels of either HDAC4 or PDCD4 were assessed using Western blot. And for the Western blots, tubulin was used as a control. And as you can see, the protein levels of either HDAC4 or PDCD4 in cells transfected with the miR1 mimic were reduced to that of the mock levels through at least 72 hours, suggesting the effectiveness of miR1 in regulating the protein levels of HDAC4 and PDCD4. And to turn to an experiment done with miScript microRNA inhibitors, HeLa S3 cells were transfected using the high perfect transfection reagent with inhibitors for miR15, miR16, miR21, and miR25, or an inhibitor with a scrambled sequence, and this is the negative control. At the time points indicated, luciferase reporter constructs with the appropriate microRNA binding sites were transfected. And a reporter construct that is not regulated by any microRNA was used as a positive control. And 24 hours later, luciferase activity was measured. Through 48 hours, there is 100% inhibition of microRNA activity for miR15A, miR16, miR21, and miR25 as measured by luciferase activity. Even at 96 hours, there is sufficient inhibition of microRNA activity, demonstrating the effectiveness of the microRNA inhibitors. And for both miScript microRNA mimic and inhibitors, QIAGEN offers a variety of controls, including mimic negative controls, mimic positive controls, inhibitor negative and positive controls, and transfection controls. And we really highly recommend using these transfection controls in every experiment. I'd like to now conclude with a couple resources that will be very helpful in your experiments. The first is GeneGlobe, and this can be found at QIAGEN.com/GeneGlobe. And this site is a very good site to explore and order gene and pathway-specific products for a variety of applications, including gene silencing, microRNA research, protein research, and a variety of other applications. And to use this, you simply enter your search term of interest, such as miR16, and you can then select your species. You can either select all, or you can narrow your search to your species of interest. And what will result is all the product offerings QIAGEN has for that specific microRNA. And finally, the last slide I'd like to show is really the new microRNA detection portfolio and workflow available from QIAGEN. From purification using either the microRNAs or PAXgene kits, through analysis where you can do profiling and individual assay quantification with a microScript PCR system, and finishing with functional studies where you can use microRNA mimics, inhibitors, and target protectors to elucidate your microRNA function. QIAGEN has a solution for you. In addition, I'd like to again mention that the purification is amendable for automation using the QIAcube, and we also have a very good qPCR instrument known as the RotorGene Q to do your real-time PCR studies. And with that, I'd like to conclude and take any questions that you have. And again, thank you for your attention, and please listen to our upcoming webinars, again, that will occur over the next couple of weeks. Thank you very much.